And I'll start this morning session, the second morning session, by saying Assalamu alaikum to you. Assalamu alaikum. I do understand that आप 2 दिन से बहुत hectic जो है ना मसरूफियात में शामिल हैं. We are running 6 and 7 parallel sessions since yesterday, and today we also had 7 parallel sessions. तो मुझे अंदाज़ा है कि it's a bit tiring now, but still I am going to actually initiate one of the most important lectures of King Edward Medical University. और इस succession में हम कर रहे हैं just to remember one great individual, one great Kemcolian, Mr. Ayub Ommaya. So इसलिए मैं चाह रहा था कि मोइन ने इसलिए मुझे रोका था कि अभी शुरू ना करें, because we wanted our students to be here and well seated so that we can start with this. So I'll once again start by saying Assalamu alaikum to you again. Thank you very much. But for me it is a great pleasure that my very own teacher, जिनसे मैं पढ़ा 3rd year 4th year final year में, उनको हमने दरख्वास्त की कि जिस शख्सियत के बारे में हम बात करना चाह रहे हैं आज, जिनके बारे में जिन्होंने बात करनी है वो इतने अहम है कि उनकी साइटेशन, जिसने आके बात करनी है उनकी साइटेशन, मैंने कहा मैं उनकी साइटेशन भी मैं खुद पढ़ूंगा बिकॉज ही इज़ अ ग्रेट साइंटिस्ट हिमसेल्फ. तो आए, लेकिन मेरी बड़ी खुशी और एक तरह से अकीदत की बात है कि प्रोफेसर जावेद अकरम साहब यहाँ पे आज तशरीफ़ रखते हैं, जिनसे मैं 3rd year 4th year फाइनल में बैठ के इनसे पढ़ा और इनसे इल्म हासिल किया, इनसे मुस्तफ़ीद हुए, और आज तक हम जो कुछ भी हैं ऐसे ही उस्ताज़ा की रहनुमाई और इनकी जो है ना मेहरबानियों के बायस यहाँ पे खड़े हैं, जो कि आपको ज़िंदगी में हर कदम पे आपको अपना मशवरा देते हैं और आपकी हर खुशी गमी में शरीक होते हैं, ख़ास तौर पे आपकी हर खुशी को सेलिब्रेट करते हैं आपके साथ मिलके. सो सर, आई एम सो ग्रेटफुल दैट यू चोजन एंड ऑन आवर वेरी लेट रिक्वेस्ट दैट यू शुड जॉइन दिस एस्टीम्ड लेक्चर. सो थैंक यू वेरी मच फॉर बीइंग हियर. थैंक यू. ऐसे नहीं, मैं चाहूँगा तालियाँ आप उस तरह बजाएं जिस तरह आप उस्तादों का खैर मकदम करते हैं. So thank you very much. So we will embark upon, without wasting any time, we'll embark upon the greatest lecture which we are going to hear in all these scientific sessions. और जिसका मैंने आपसे जिक्र किया कि Professor Ayub Ommaya was one of the greatest Kemcolians that King Edward Medical University has ever produced. So it's worth, listening about him. And I will invite Professor Ali Javed for actually come and talk about him and give his Ayub Ommaya lecture. Lekin before delivering this lecture, I would like you to get introduced to Dr. Professor Ali Javed. Can I have Professor Ali Javed? So Professor Ali Javed is seated in front of you, and let me introduce him to you, who is going to talk about Professor Ayub Ommaya. Professor Ali Javed is the professor and head laboratory of translational research in neuropsychiatric disorders. ये मैं थोड़ा सा आपको बता दूं कि neuropsychiatric disorders होते क्या हैं. It's about a research center studying brain and mental health diseases in the Łukasz Research Network, Poland. मैंने इसको ठीक प्रोनाउंस किया, बड़ी कोशिश है क्योंकि बिकॉज दिस इज़ प्रॉब्ली इन पोलिश, तो मैंने बड़ी मुश्किल से सीखा कि इसको Łukasz जो है ना रिसर्च नेटवर्क पोलैंड से ये इसके साथ एसोसिएटेड है. प्रोफेसर अली जावेद इज़ अ रिनोन्ड फिजिशियन साइंटिस्ट. मैं फिर आपको बता दूं कि फिजिशियन साइंटिस्ट क्या होते हैं. दीज आर डॉक्टर्स हु कंडक्ट साइंटिफिक रिसर्च, ट्रू हार्डकोर साइंटिफिक रिसर्च. हिज वर्क फोकसेस ऑन न्यूरोजेनेटिक्स of childhood trauma, memory and neurodegenerative disorders. और न्यूरोएपिजेनेटिक्स भी थोड़ा सा मेरे लिए भी थोड़ा सा नया लफ्ज़ था, तो फिर मैंने सोचा कि ये क्या है, तो उसके बारे में पढ़ा तो ये कहा कि न्यूरोएपिजेनेटिक्स इज हाउ लाइफ एक्सपीरियंसेस अफेक्ट ब्रेन फंक्शन थ्रू चेंजेस इन जीन एक्टिविटी. सो दैट इज व्हाट ही एक्चुअली डज मोस्ट ऑफ द टाइम, एंड ही रिसीव्ड एडवांस्ड रिसर्च एंड क्लिनिकल ट्रेनिंग फ्रॉम ETH Zurich University, which is the leading, which both of them are the leading universities in Switzerland, and from the Baylor College of Medicine, USA, which is a top medical research university. He has authored more than 80 publications. They're all hardcore scientific papers in leading journals such as Nature, Neuroscience, and Neuron. With a current H index of 33, a measure which shows both productivity and impact of the research work. Professor Javed is a FENS Kavli Scholar, which is a member of a prestigious European Neuroscience Network for outstanding young scientists, and is recognized among the world's top 2% scientists by the Stanford University, which is a rank based on research impact and citations. So ladies and gentlemen, let's put our hands together to welcome Professor Ali Javed.
Thanks a lot, first of all, Dr. Ayaz, for this very kind introduction. Assalamu alaikum and good morning, everyone. Well, actually, good afternoon, everyone. It's an absolute honor and privilege to be delivering the Ommaya lecture. When I got the invitation from Dr. Shafiq, and again, thanks a lot, Dr. Shafiq, for the very kind invitation. The first thing which I thought was it's probably a scam, because how could I be invited to deliver the Ommaya lecture? I mean, we are talking about somebody whose name is synonymous with cutting-edge technology, something which was, a revolutionary breakthrough in the field of neurosurgery. And Dr. Ommaya was a true physician scientist and an inspiration for several of us. From Aga Khan University, I'm not a Chemcolian. Actually, it's my first time in King Edward's, but that's how I used to recognize King Edward's by the name of Dr. Ommaya. So it's an absolute honor and privilege to be delivering the Ommaya lecture. And just like him, I'm also a physician scientist. Still learning, still in the process of learning and acquiring new knowledge. And one of these very fascinating things, I got interested in during my PhD studies, and then I've carried it on for the last 10 years, is the field of epigenetics. And as you would see over here, the epigenetic legacy of trauma, a paradigm shift in the concept of human heredity. This is something which I came to know, and it was very startling, very surprising that our experiences can actually be embedded in our epigenome, can actually be passed from one generation to another, and something which can be carried on. It's actually something which is contributing to human heredity and the transmission of disease risks. It's a complete paradigm shift. It's a complete game changer. So over the next 40 minutes or so, I'll be talking about that. But what I also want to introduce and what I want to sensitize you guys about is that this topic is not something new. One way or another, humans have been very aware of the fact that what we experience or what our parents experienced or what our ancestors experienced, it is shaping us. It's just that we didn't know that this actually had a very strong biological imprint. And what you would see right now is that these things can actually be even reversed. And it has so many implications, not only for the clinics, but also for humanity. So I will start with two pieces of art, actually, not science. I'm one of those scientists who starts his talks by showing art. And I'll be playing two video clips. Now, several of you might have actually heard these songs before, but let me give you a little bit of a background and sensitize you to this topic by sharing those pieces of art. This is actually a song back from 2013, a Belgian rapper. It became very famous, so several of you out there, several of the millennials out there have probably heard it. And people thought that it was, just like a rap song with probably some kind of symbolism in there. But what actually don't realize is that this was a song Stromae, the singer who wrote that song, actually wanted to describe how living his life with an absent father has transformed him into becoming an absent father himself. So he acquired from his father this, this trait that he was not always there for his children. And he always wondered, why is this happening? So this song was about this, transgenerational, from one generation to another transmission. And what's also very interesting is that the reason why his father was an absent father was because he was serving in the military. He was actually involved in this genocide in Rwanda. And eventually, he also perished. He passed away due to the genocide. Now, there's another song with a lot of you Gen Zs out here, the students, the Gen Z students would be very well versed off because this was very popular last year. Okay, you listen to the song and you think that this is just a song about somebody rambling about their ex, but it's not that. This is actually a song about transgenerational trauma, and let me actually tell you that I've spoken to Charlie XCS, the singer of the song, like we have spoken, and she said that actually this song was about transgenerational trauma, how this affects people's behavior, how they behave in relationships, because they also acquire some traits because of the patterns. And actually, if you look at the lyrics, the apple is rotten in the core because that has been passed on from the apples before, So it's like one apple leading to this sort of transmission to the other apple. So this was also a song about transgenerational trauma. This figure which you see over here, it's actually from one of our reviews which we wrote a few years ago. And what we found out was that different kinds of exposures in parents. Now these could be positive exposures such as exercise, but mainly a lot of negative exposures, what we call as risk factors, be it metabolic disease or obesity or demographic risk factors such as aging or psychological trauma or stress, toxicants such as smoking. All these exposures are associated with transmission of disease risk in the next generation. In this review, we looked at thousands of different studies, and we were able to pinpoint that several of these factors, especially psychological trauma as well as metabolic disease, were very strongly associated with transmission of disease risk in the next generation without presence of any mutation. So this was not a genetic transmission. This was not this familial transmission which you see in diabetes, hypertension, or with different kind of mutations, but this was an actual potentially epigenetic transmission. And how I learned about all of this was actually studying, doing my MD PhD in a lab, which I was crazy enough to decide to do after my clinical training in neuropsychiatry. And this was our landmark paper in Nature Neuroscience, where we first showed the world the evidence of this epigenetic transmission through a study on mice, a model of impaired maternal care. We call this model MSUS, which stands for unpredictable maternal separation and unpredictable maternal stress. Is there a pointer, by the way? That would make it very helpful if I'm able to guide the students, yeah. So while we get that laser pointer, let me explain what this model is actually about. So what we are trying to do here is to replicate childhood neglect or childhood, trauma or childhood abuse, which is very common, perfect, yeah. So what we are trying to replicate here, what we are trying to recapitulate here is this childhood abuse or childhood neglect, which is the single most important risk factor for childhood, adolescent, and also adult neuropsychiatric disorders. And in this model, the newly born pups, they are separated from their mothers, for 3 hours daily in an unpredictable fashion. During the same time, when they are separate from the mothers is exposed to different kind of stressors. What we are showing over here is this cold swim stressor, where mice, when they are put in cold swim, it is a very stressful experience for them, increases their cortisol, epinephrine, all of that. And then this dual hit leads to these mice who have been traumatized early in life to develop a multitude of different phenotypes, or what you may call symptoms. And those symptoms can range from changes in their behavior, like, which I will explain in more detail in the next slide. But also we see changes in their immune functions and their metabolism. But perhaps the most striking part was that when these traumatized mice, when they are mated with naive mice, which were never exposed to any kind of trauma, what we call wild type mice, then we see that even the offspring has all these different phenotypes. So this is transmitted from one generation to another. These are not transgenic mice, they are not genetically modified, but just the parent was given a certain exposure, and it is leading to a phenotypic transmission to the next generation. And in fact, this goes on up to 4 to 5 generations of mice. So the question which popped up was, what is leading to this kind of transmission? So before that, just let me allude a little bit to the phenotypes we see. So we generally see that these mice have a complete alteration of their nature. Mice are nocturnal animal. They like to spend time, in the dark. So if you would put a light and dark compartment, the mice would generally stay in the dark. They only go to the brightly lit compartment to see if there's any kind of food or any other reward or stimulus there. When they don't see a reward there, they come back to the dark compartment and they spend most of the time there. Now, if a mouse is spending too much time in the brightly lit compartment, it means that they are taking unnecessary risk, which is, again, something which we see in human psychiatric disorders like borderline personality, that people are, indulging in risk-taking behaviors. If the mice would never come to the brightly lit compartment and just stay in the dark compartment, then it means that they are increasingly anxious. And what we see in these MSUS mice, but also their offspring, is that they spend a lot more time in the brightly lit compartment. So they are showing these risk-taking behaviors. Another phenotype which we see here is that when we test them for these pro-depressive behaviors, they also show indicators of depression. So how we check for despair or pro-depressive behaviors in mice, we place them in this beaker of water. Now, a mouse which has not been exposed to any kind of trauma or which is not showing this depressive behavior, they would try to escape. But the mice which are showing this pro-depressive behavior, they keep on floating in water. This is very similar to how, humans, they become they have these features of anhedonia. They start, they kind of start giving up on life when they are, showing these pro-depressive behavior. This is exactly what we see in mice. Besides that, we also see changes in their social interactions. We see changes in their spatial memory and navigation skills. And also importantly, we see changes in their metabolism. We saw a significant decrease in their high-density lipoproteins, HDL levels over there. So these phenotypes start appearing in mice as well as their next generation. And what we wanted to figure out was, is this transmission from one generation to another, is it biological? Is it germline dependent? And also, is it genetic or epigenetic? So we performed this very elegant study and our landmark study back then, where we took the RNA from the sperm of the traumatized mice. So not DNA, because we didn't want to go for the genetic transmission, but epigenetic transmission, post-transcriptional. So we took RNA from the sperm of the traumatized mice, injected it into the one-cell embryo formed from the mating of naive mice, and that led to recapitulation of all the different phenotypes. Yeah, so this is very similar to in vitro fertilization, just that more than in vitro fertilization, we also took the epigenetic material from the sperm. And what that actually meant is that trauma initially perceived by the brain leads to certain kind of changes first in the brain, and they are somehow carried on to the germ cells. They are already getting imprinted in the sperm, and when you take the RNA of the sperm, inject it into the embryo from non-traumatized mice, it still recapitulates everything. So now the link between the brain to the reproductive organs and the germline was already established. So the next question for us was, okay, what is the connection between the brain and the reproductive organs for this epigenetic transmission to the next generation? Now, very intuitively, you would think that, okay, what's connecting the brain to the rest of the body are like, blood, right? Like circulation. And then, of course, in science, we need to validate everything through like very strict experimental procedures. So what we did was we performed a chronic transfusion experiment. So we took the blood from the traumatized mice, and then we longitudinally did a transfusion of the serum in wild-type mice, in naive mice which were never exposed to any kind of trauma. And this led to a recapitulation of the whole epigenetic signatures in the sperm, like, of the non-traumatized male mice which had received the transfusion, which were the recipient, as well as a recapitulation of the phenotype in the next generation. So now what that means is the trauma initially perceived in the brain is leading to changes in the blood. Blood becomes the mediator of that between, like, the brain and the reproductive organs. The effects get transcribed and imprinted in the germ cells, which then carry it on to the next generation. So this opened up two more questions. Number one, blood contains hormones, cytokines, circulating non-coding RNAs, like many different factors. So what is the mechanistic hub responsible for induction of these germline changes? And of course, most importantly, what is the relevance of all this for humans? So this was the challenge which we took up in my lab like about 5 years ago. And when you want to study something like that in humans, there are several challenges, several important points which you have to address. Number one, complexity and face validity. What I mean by face validity, is this mouse model the relevant model to recapitulate human traumatic experiences? And then what we realized was, no, okay, maybe it's a good model to study childhood neglect, but when we think about traumas which are presently happening in the world, it's a lot more. It's actually wars, genocides, so many things happening in the world, so we need to study actually humans or create models which have better face validity. What about body fluid-based transmission? So in the mouse study, of course, we could take this, like, the RNA from the sperm of the traumatized mice, but when we would look at natural human birth, then of course there is the importance of the vaginal microbiome during the vaginal delivery. What about, post-birth exposures such as milk, lactation? Finally, what about the biological versus psychosocial transmission, right? I mean, you could just assume that humans are, emotionally a lot more complex and a lot more mature than mice. So what about the psychosocial component of that? What about genetic heterogeneity? When we study something like that in mice, we have, all the mice from the same strain. They are genetically a lot more homogeneous. They are inbred, but humans, can be genetically a lot more diverse. And finally, we wanted to study this in humans, but we didn't want to do a correlative study. We wanted to actually have strong mechanistic and causal underpinnings. So we strategized. We came up with a multi-modality investigation model where we assembled human trauma cohorts around the world, where we are still studying our mouse model, but we have refined it to make it more recapitulative of what we find in humans. And then we are using a combination of samples derived from humans, but using them to stimulate, to study very innovative models like organoids. Let me ask a question to the students here. You can just raise your hands. How many of you know about organoids? Okay, so let me tell you simply, organoids is a stem cell technology. So you can take the skin fibroblasts, reprogram them into induced pluripotent stem cells. In the end, we are all formed from the embryo, right? So we convert it into this induced pluripotent stem cell, which is like the one-cell embryo, embryonic stem cell. And from that, under the right conditions, you can grow any organ. So I have grown my own brain in a Petri dish several times. You just need a punch biopsy and do that. And these things can be implemented, and hopefully, inshallah, one day you guys will be working on these models in Pakistan, I'm sure. All right. So let me start with, our biggest effort. And we assembled different human cohorts, human adverse childhood experience, like childhood trauma cohorts, across the spectrum of genetic heterogeneity. This world map, which you see over here, it's a very unique map. This is actually one of the maps where Pakistan is #1 in the world, because this is the world map of consanguinity. So we decided to assemble cohorts across the spectrum of genetic heterogeneity, or what you call like consanguinity, with the sole purpose that if you want to single out the effects of epigenetics versus genetics, you want the effects of genetics to be minimum. And for that, we needed to have these cohorts across the spectrum. So the first cohorts we assembled were in Pakistan, which has the highest rate of consanguinity in the world. So let me tell you that if I would just do a genomic sequencing on all of you, you would all turn out to be distant cousins of each other, one way or another, because of the cousin marriages over hundreds and hundreds and hundreds of years. They have led to a lot more, genetic consanguinity and genetic homogeneity compared to Western populations. So we assembled our first cohorts in Pakistan, and our cohorts, the first cohort was children with paternal loss and maternal separation, which we assembled in collaboration with the SOS Children's Village. This cohort is of children who lost their fathers within the preceding year. The mothers are alive, but they are financially destitute. They were not able to raise the children on their own, so they gave their children for adoption to the SOS Children's Village. The children are between the age of 7 to 12 at the time of the study, so they were emotionally mature enough to understand that they have been separated from their mothers. So this model, this cohort, was recapitulative of this MSUS model of maternal separation, maternal stress. Here we also had maternal separation, and the maternal stress component was coming from the paternal loss, the loss of the spouses for the mothers, which was a very stressful scenario for them. But of course we wanted to study the sperm samples, and for that we needed also adult cohort. So in collaboration with the Chughtai Labs network, we actually assembled a cohort of adult men, and we collected their seminal fluid, and then we retrospectively analyzed them for simple and complex childhood trauma. These are the first cohorts. Then we assembled a cohort in Bosnia, which on the consanguinity map is somewhere between Pakistan and the Western population, so they have mid-level consanguinity. And there we assembled a cohort, and this is actually, to my knowledge, unprecedented. This is the only cohort in the world where we have studied across generations the epigenetic signatures as well as the germline. This cohort is also very unique because these are the survivors of the Srebrenica genocide in 1995. Srebrenica genocide, I would say before Gaza, it was probably the most unfortunate event in human history, where around 9,000 individuals, they were killed over a period of 2 weeks. So these were ethnic Bosnian Muslims. They were in a United Nations shelter. When the Serb forces came, the UN peacekeeping forces, it was the Dutch platoon, which actually abandoned them. They ran away. So these people were taken as hostages. And what they did was that they separated the adult men, the fathers, from the mothers and the children. Then they separated the women and girls. They were sent away. They were sexually exploited. There were a lot of really horrible things happening over there. But what they also did was that they made the boys watch their fathers being killed in front of them, one after the other. I know it's a very heavy topic, but it's important to give you also a historical context of it. So what we are studying are those boys who saw their fathers being killed in front of them, and they themselves are fathers now. So we approached them, we convinced them to become part of our study by saying that this is something which will help us, the population which are affected by the genocide now, for example, the Palestinians. And they were brave enough and courageous enough to be part of that. So we collected the sperm samples from those men who experienced this genocide trauma during their childhood. And we also collected the samples from their own children. And finally, our last cohort, which I will talk about, is the mother-child dyads, where there was a history of childhood trauma in the mothers. And the uniqueness of this cohort is that here we have collected human breast milk samples. In fact, I will start with that because this was our recent study, which got published in Nature Translational Psychiatry just a couple of months ago, and it has been also very popular in the media, especially in Western media. So this study is about epigenetic changes in human breast milk of mothers who had childhood trauma and how it is impacting the behavior of their offspring. The study actually started at the time of delivery, and it was a completely blinded study. At the time of delivery, we approached different mothers and we asked them to become a part of the study, where we said that we will see the influences of their life exposures on the behavioral trajectories of their children up till 1 year of age. And then we did some kind of a filtering mechanism. We called them again at 5 months, and then we asked the women which of them have exclusively breastfed their children. Then our cohort became more filtered and cleaner, and we were able to select only the parent-children diets where the mothers were exclusively breastfeeding their children. At that time, we did a thorough analysis of the behavioral trajectories of the children. Actually, the behavior of the children can be studied even at 5 months. There are very validated scales for that. And then we studied the behavior of the children again at the age of 1 year. And then at that time, we asked the mothers about their own childhood traumatic experiences, so not to bias them in any way. It was a very comprehensive study, but let me highlight the key findings. We found out that certain epigenetic factors we call as microRNAs, which are important for gene regulation at the post-transcriptional level, we found them to be significantly increased in the breast milk of mothers who had high childhood trauma exposure. We also found out that there were significant correlations between the severity of these childhood traumatic experiences and the expression of these epigenetic marks in the human breast milk. So we were wondering if it is possible that this is confounding by postpartum depression. Is it possible that women who had childhood traumatic experiences, they had more postpartum depression, and that was somehow affecting the molecular composition of the milk? But this did not turn out to be the case. We found that the association was with childhood trauma, not postpartum depression, either in the expression of these epigenetic factors as well as their correlations. However, the most important part was that this had a very strong correlation with offspring temperament. And what you see over here are correlations with high intensity pleasure in children and expression of this particular microRNA in the breast milk. So you could see that the social and adaptive behaviors of the babies already at 5 months of age and 1 year of age were determined by the presence of certain epigenetic factors in the mother's milk. So what that means is that traumatic experiences can alter epigenetic factors in human breast milk, which will then alter the behavior of the children already at a very, very young age. Now, a lot of people ask me, does that mean these children will develop any pathology like autism or ADHD? At this point, we don't know. We just know that their behavior is different, but we are longitudinally following them up to see if they will have, if they will develop a particular pathology or not, or if they will have any kind of resilience against these pathologies. But what we see is that definitely there is this epigenetic transfer through the human milk. So the interim conclusions are that adverse childhood experiences are associated with characteristic microRNA and metabolic signatures in human breast milk. These are correlates of early temperamental trajectories. The study, of course, does not account for several other factors like paternal influences, oocyte-based transmission, vaginal microbiome. This is why this is not causal, but correlative at this point. But definitely what it means is that we have identified a biomarker, a non-invasive biomarker, which can predict altered behavior in the children. So we have a potential biomarker for this intergenerational transmission through the human milk. Now let me get to the Bosnia study, because that is not only about biological plausibility, but has a very strong association almost at the causal levels. And I just want to explain that this study was done through I got funding from European Union. It was a huge grant, and I was able to I still have a whole team in Bosnia, which I'm co-leading as well. So we recruited families. They went through very comprehensive, strict procedures. We collected the seminal fluid, palleted the sperm out for the fathers. We collected blood samples from the mothers and the children. And we did a very comprehensive, systemic analysis of all health parameters, as well as, all the psychometric analysis of the children, as well as the parents. Let me highlight some of the key results here. So this was Srebrenica, where the genocide happened. We also had a control group in Tuzla, where the level of trauma experienced by people was a lot less. Plus, we also selected for a group which did not experience any kind of significant childhood trauma. First, we were able to confirm that there was significant childhood trauma in these men from Bosnia through 2 different scales. When we looked at their current mental health functioning through the WHO self-reported questionnaire, we also found their current mental health functioning was impaired, so they're not doing too well mental health-wise. But despite that, what we found out was that they are trying their best when it comes to being the parents. When we compared the parenting quality, and this was cross-validated, so we asked the mothers if they think that their husbands are good fathers and the other way around, we found out that the parenting quality was comparable with the control groups. So it's not that being exposed to the genocide during childhood made them non-nurturing parents. They were still quite good. But despite that, we saw that in the children from Srebrenica, from these families, there were a lot more symptoms of oppositional defiance disorder against teachers, as well as parents and peers, as well as ADHD. This was reported both from the mothers as well as fathers, and the scores agreed very much with each other. Now keep in mind that these children were not exposed to any kind of war or genocide themselves or any kind of traumas. Their parents were the one, their fathers were the one which were exposed to the genocide, and despite that, they had all these different symptoms of ADHD and ODD. But this was reported by the mothers and fathers, so we also wanted to directly analyze the children. So we used our depression and anxiety scales, directly interviewed the children, and we saw that there was a lot more depression in the children from Srebrenica families and a lot more anxiety in the children from Srebrenica families. By the way, again, all our studies are blinded. So because I'm the principal investigator, I don't have access to data. I don't analyze it myself. They're actually outsourced. So our data is analyzed by bioinformatics group in Switzerland, who don't know about the study design, and everything is done in a completely blinded fashion. Okay, very interestingly, and this was advanced bioinformatics analysis, we call it SEM, structural equational modeling, we looked at all different parental factors, and we wanted to assess which parental factors can predict the transmission of these psychological symptoms in the children. And we found a very strong association with paternal HDL levels, with the lipids of the parents. And I will try to simplify, these complicated equations which you see over here. But basically what we found out was that the trauma in the fathers predicted depression and anxiety in children. However, trauma in the fathers also led to decrease in HDL levels. To put it simply, we say that HDL is the good cholesterol, right? So we see that the good cholesterol was lower in the fathers who had this genocide trauma. Importantly, in the fathers where the HDL levels were high, we actually saw that this correlation was inverted. So high HDL in fathers was associated with lower depression and anxiety in the children. This is also explained over here. Here are the HDL levels. You see that this is the baseline, this is optimal, this is suboptimal. We see that when the HDL levels in fathers are suboptimal, they actually predict depression and anxiety in the children. When they become suboptimal, this association is converted. Then it's not a positive correlation anymore, it becomes a negative correlation. So what that means is that fathers' lipid profiles are important determinants of the children's psych scores. And then we did very comprehensive advanced transcriptomic analysis in my lab. So we found out that certain lipid-associated epigenetic factors, microRNAs which regulate lipid signaling, they were similarly altered in the father's serum as well as children's serum, and several of those were also altered in the sperm of fathers. Yeah, so you see, like, we see changes in the lipids in fathers, we see changes in the lipid-associated epigenetic factors in their sperm, and then what we also saw was that there was a very strong correlation between these lipid-associated microRNAs in the father's sperm and depression and anxiety score in the children. And prominently, this had clear gender dimorphism. We saw that the father's trauma impacted the sons more than the daughters. You would see that all these depression scores, which are showing a positive correlation with the epigenetic factors in the father's sperm, these symptoms were more prominent in the sons compared to the daughters. So this also made us wonder if this could be some kind of a Y chromosome-based transmission. Did not turn out to be the case. This is clear epigenetic transmission, but it's more prominent in the male. It's a patrilineal pattern of transmission. Okay, but then in science, like I said, when we see important observations, when we find these predictors, biomarkers, correlation, it's very important to show a cause and effect relationship. So this is what one of my PhD students has been doing for the last number of years. She is collecting lipid messengers from our mouse model of trauma. So we know lipids have nutritional value, but lipids are not only nutritional, lipids are very important from cell to cell communication. Let me ask a question again to you guys. How many of you have heard of the blood-brain barrier? Everyone, right? How many of you know that there is also a blood germline barrier? Right? Sertoli cells, they are forming this barrier in the testes, and similarly there are barriers like, around the oocytes as well. What is one entity, one biochemical entity which can easily cross the barriers? Lipids, right? So lipids have a key role also in this cell-to-cell communication because they can go across all barriers. And I always say that nature, wants us to learn, and nature wants us to propagate. And that's why it protected the brain with the blood-brain barrier, it protected the reproductive organs with the blood germline barrier. But in the end, lipids can easily transfer. So it's not surprising that lipids are also responsible for this communication from the brain to the body to the germline. And this is what we actually checked by collecting all these lipid messengers, which included extracellular vesicles, which are protected by this phospholipid bilayer, high-density lipoproteins, which I already showed that they are altered also by traumatic experiences. And we did a very interesting cross-injection study in mice. So here we have traumatized mice. We took their lipid messengers, we injected them into control mice, and then we checked if the next generation would have transmission of the symptoms. Similarly, we took the lipid messengers from the control mice, injected them into traumatized mice, and then we checked if there would be a reversal of trauma-induced symptoms in the next generation. And this is exactly what we found out. We found out that just by injecting the lipid messengers from the control mice to the traumatized mice, we can prevent the transmission to the next generation. And just by injecting the lipid messengers from the traumatized mice into the control mice, we can induce the transmission. What that means is that lipid messengers are the key. Lipid messengers is the way one generation communicates to the other by inducing these changes in the germline. What we also found out, and this is something very fascinating, don't focus on like the heat maps and stuff, I will explain it very simply. So we found a number of changes in the epigenetic factors in the sperm after trauma. We found in the serum of children, in the sperm of adult men, as well as the human breast milk. These are Polish mothers, these are Pakistani children, these are Bosnian men, right? Interestingly, what we found out was that the signatures of trauma are very similar across body fluids, across ethnicities. So Again, 3 different ethnicities, different age groups, and we always saw an increase in this microRNA 2233P. This was increased in the Pakistani children with trauma. This was increased in the Bosnian men with trauma. This was increased in the Polish mothers with trauma. So essentially, what we believe is that we have identified a biomarker of childhood trauma, which will be the first psychiatric biomarker, which can also predict its transmission to the next generation. What we also did is we did a study in mice where we did embryonic manipulation of this particular microRNA. So we altered this microRNA in the embryo. We did the embryo transfer, created new mice, which had altered miR223 already in the embryo, and we followed them for up to 1 year, and we did an analysis of their brain as well as their metabolic signatures. And we found out that even after a year, when we did this embryonic manipulation, this microRNA persisted in the brain, and there were changes in the metabolites. And just read the metabolites: phenylalanine, tryptophan, phenylalanine. Again, these are all the different metabolites which are important for neurotransmitter synthesis. So now we also have biochemical signatures for how the behavior, these pro-depressive behavior, and how all of that can be altered through these epigenetic factors. But so far, whatever I've showed you is from different cohorts. And one question which always popped up was people asking me that, okay, you have cohorts, but they are smaller cohorts. You had 100 mothers, you had 80 families. So can you show us some data that this phenomena of transgenerational transmission, it exists at a wider level? So for that, we did a very interesting bioinformatics study, which offers big data perspective. And we looked at historical adversity and prevalence of mental health metabolic disorders in the United States. We focused on a period of 1929 to 1945 because this was a period of adversity in the United States. There was the Great Depression of the 30s, and then there was the Second World War. And we used socioeconomic status as a marker of adversity, that the American states which were poor at that time had lower socioeconomic status, they had more adversity compared to the states which had higher per capita income, which had higher socioeconomic status. And very interestingly, we found out that the socioeconomic status across US states in 1929 to 1945 predicted the prevalence of diabetes as well as significant mental illnesses. So mental illnesses which required therapy or hospitalization 65 years later, from 2010 onwards, So what that means is that 65 years is a significant gap, so we are not looking at the same generation, but we are looking at potentially the next generations. So historical adversity is leading to countrywide changes in the prevalence of diabetes and mental health disorders in the United States 65 years later. We also found out actually, and this is a very startling observation as well, that the states which had the higher per capita person income during 1929 to 1945, or in other ways you can say that the states which were the richest, now their prevalence of diabetes is lower than the mean, and the states which were the poorest during 1929 to 1945, now their prevalence of diabetes and significant mental illnesses is much higher than the median. So what that means is that historical adversity could have impact on the health demographic of states several years later. So what are the implications of this for all of you out here, mostly physicians? Well, many. If actually indeed lipids and lipid messengers are important for the long-term consequences of childhood trauma, and they can impact the germline, which can lead to transmission of disease risk to the next generation, then it means that through simple lifestyle modifications involving lipid-modifying interventions, this can be prevented. This also means that there should be a bigger focus on contribution of fathers' mental health, in addition to the maternal mental health, also to the disease risk. We often think that whatever the child inherits, it is related to how mothers, like what mothers experience during their pregnancy, but there's a very important contribution also coming from the father's side to the epigenetic risk of diseases in the next generation. What it also means is that through simple lipid modifying interventions before conception, perhaps these risks could be mitigated. Finally, when you go to the clinic, you always take history of diabetes in the family, history of hypertension in the family, cancer in the family, but maybe it's also important to start taking history of exposures in the parents. Perhaps traumatic exposure in the parents can also predict disease risk in the next generation. So all of you medical students out there, start doing it now. Maybe you will also gather, you may be able to collect some very interesting data which you can analyze and find more associations. And of course, the implications of this work, they go beyond the scientific world, they go beyond clinic. I mean, just look at the world around us. You know what's happening in Gaza, you know what, the war has been going on in Ukraine for a number of years. So it has been like a mission for myself, as well as my lab, that we want to highlight that the concept of transgenerational trauma is real, it's biological, and all the children who have been suffering during the wars and genocides, not only will they suffer themselves, but potentially generations and generations of humanity have been scarred by whatever is happening. So I've been writing editorials for, you can see the journals, Nature, some of the best journals in the world. We have written editorials and reviews also to highlight this part. And with that, I will come to the end. Before you start clapping, I just want to introduce all the research which our lab is doing. I'm very, very open to collaborations if approached in a meaningful way. And of course, these are conversations I would love to have with the faculty and the students at King Edward's. And of course, usually, when you go to a lecture, they end a slide with acknowledgements and people's names are written over there. But we decided in my lab that a nice way to pay tribute to my lab will be to highlight everything which we work on and introduce the whole team to you. So we have a little video presentation, and right after that, I'll be happy to take any questions. Thank you very much.
My dear students, it's a great occasion for us that you have been an audience and a visual beneficiary of this great lecture. And I would actually really invite our students to actually ask questions, some intriguing questions from the presenter, so that you can really benefit from, because it's a rare occasion that a lecture of this level has come from an alumnus of Aga Khan Medical University to King Edward Medical University. So please do ask questions. और एक मैं बात आपको इन्होंने बताया ना कि this epigeneity is one of the main reasons for this epigeneity is consanguinity. Do you understand what is consanguinity? क्या होती है consanguinity? Cousin marriages. So my message to all these ladies sitting here is, don't marry your cousins however good looking they are.
As decided speakers, splendid presentation, and from epigenetics. But the thing is that at the first glance to the research and the study, the first thing that comes to my mind is that there are so many confounders there. Like the definition of the word childhood trauma. How do you define that? I mean, the examples that you quoted of Serbia, of Gaza, of course there are, definite, you see the trauma, but the definition of trauma would be so broad. And secondly, you said that the biological transmission was due to HDL. So did we know the HDL levels prior to exposure to trauma?
Yeah, excellent question. So in terms of like childhood trauma or childhood adversity, I mean, so there are two ways to look at it. One is, of course, like we rely on the scales, right? The childhood trauma questionnaire, like Harvard trauma questionnaire. So these are like standardized validated scales. And they define a threshold for like, when the exposure to adversity starts becoming a risk for psychiatric illnesses in the long run. So this is our criteria which we use in the human studies. And in the animal studies, we try to recapitulate the human setting. Just like you saw in the MSUS model, we combine maternal separation and unpredictable maternal stress because that leads to childhood neglect in human conditions. So we try to recapitulate that. Of course, there is a whole variety of these things. We have also looked at that. Like I'm not showing like all the data, but we have also looked at which kind of experiences, which kind of traumatic experiences are subjectively considered more severe. And it turned out that, for example, like, when we look at parental separation, parental death, bullying, like all of that. So of course, we found out that assaults, sexual abuse, sexual assaults, they were subjectively considered more traumatic. And then we also see more effects on the germline level, and then also in the transmission to the next generation. So it depends on the subjective severity of trauma. With your other question about HDL, so HDL is one of the key lipid messengers because it carries these epigenetic factors from one generation to another. But it's not only HDL, it's also other lipid messengers, for example, extracellular vesicles, which have this phospholipid bilayer, and they also transmit. In the study, the study which we did in Poland, the milk one, there we have both the analysis of pre-exposure HDL levels, and then we have at the time of pregnancy, we even have the, during lactation, the HDL levels. The study in Bosnia, the study design, it kind of precludes us to understand what was the HDL level before conception because, at the time when there was war going on, there were no medical assessments, and we only assessed their HDL, before the time of sperm collection. But we are longitudinally following them up. And what we have also started is a treatment trial. So now, all these men in the Srebrenica cohort, we are putting them on a lipid modifying intervention where we are giving them ultra omega-3 supplementation and 10,000 steps every day. And we are monitoring how, by improving their HDL levels, if we'll be able to mitigate the epigenetic marks on their sperm. And that will give a conclusive answer. But very, very important questions, yeah.
About, you said you graduated in Karachi, your first time coming to Lahore. So welcome to Punjab, and the amount of generational trauma we have in Punjab, you won't find it, and you won't have to go to Poland or USA or anywhere. So if my grandparents would be alive, there was a comment they used to say, just summarize your lecture by saying, it's sort of a misogynistic comment, but it might have been relevant at that time, that Kudi nu na maro, kudi ki maa nu maro. That was one of the quotes people used to say. Yeah, munde nu na maro, munde de pyo nu maro. ऐसे कर लेते हैं. मेरे ख्याल में आपका लेक्चर सुनने के बाद उस कोड को शायद वो हमारी जो जनरेशन है वो चेंज कर दे और वो कहे कि कुड़ी nu na maro, तो मुंडे nu भी ना मारो, तो the pyo nu na maro, तो दिमाग भी ना मारो. Because all this trauma is going in generations. So my question to you is that this is more around looking at the patterns and finding what epigenetics can do, and it runs in generations. What sort of interventions you are looking forward, or you're collaborating with your colleagues internationally to look for some sort of novel interventions? It could be social intervention, or it could be even drug intervention, I'm happy to take a portion of few milligrams of a trauma tablet, maybe, if it's developed in future. Please.
Thank you. Thank you also for bringing this, like, very important, like, social and cultural context into this. And first of all, ji, Lahore is home. I was, my mother lives here. So मैं AKU में गया था, but I was living in the hostel, in the dormitory over there. So Lahore, I have a very strong connection to. And of course, like what you're saying, interventions, this is where we are at right now. Like, initially when I started working in this field, like from 2014 onwards, it was still during my PhD studies, the first thing was to actually found the cause and effect association in mice, then translate into humans to have predictive biomarkers and to see if the phenomena exist in humans. And now where we have everything figured out, we have already done the preclinical studies in mice, right? That's where I showed that by the, this injections of lipid messengers, we are able to either mitigate or also induce, right? So we have all the pathways figured out. Now we are working on two angles. One is like a simple lipid modifying intervention trial, which we are doing in Bosnia. And actually the results will be ready in about 6 months. We also have ethical approval from the Bosnian Health Ministry that we will be able to acquire more families affected by the genocide, and we will be able to follow them up for 6 years to see now after the intervention if we are able to prevent the transmission to their second child. First child, we see that they are impacted, right? So this will be like, I would say, the most conclusive evidence of that lipid messengers are important. The second is RNA therapeutics, right? So we found this microRNA 223, and we already showed in mice that by doing the embryonic manipulation, we see these changes in the brain and in the metabolites, neurotransmitter synthesis, all the way up to 1 year. So that means that can we also do some kind of targeting through these microRNA-based therapeutics? This is something which I can envision will be there, but this will probably take much longer because it, of course, requires a lot more approvals and a lot more procedural regulation. But yeah, I see lipid modifying intervention and microRNA therapeutics. That's the way to go.
Thank you. Sorry. Can I ask? I was wondering, you were looking only at these perhaps of the parental trauma that we are talking about is coming from the male side. And it has been postulated that most of the child's intelligence comes from the mother because of the maternal RNA, which is only present in the ovum. And the human milk, we know, it responds to the way when the baby suckles, the saliva transmits to the nipple and the mother's body responds, and it changes the secretion of the milk which is coming to the mother's breasts. So how can we really look at the maternal trauma which is being transmitted to the children, and is there any other way in which if we can modify the way this maternal milk is secreted which can result in preventing that transgenerational? Thank you.
Absolutely. Again, very important question. So the only reason, to be honest, we have focused on the, for the human part on the male germline is because we have access to the seminal fluid. But for studying this in, the matriline transmission, we need oocytes, right? And oocytes we cannot get. I mean, I am actually right now in a collaboration, with a gynecologist, and they will provide us oocytes, but that will be from women who get ovaries removed, usually because of PCOS, so they already have a disease condition. So for male, the accessibility is easier, and that's why we are focusing on that. And for females, that's why we focused on the milk part, because milk we could acquire. We have studied transmission, the matriline transmission, female transmission in mice, and I can tell you that biomarkers are very similar. Same kind of pathways are also impacted there, but there are two main differences between patriline and matriline transmission. In patriline transmission, there is de novo spermiogenesis, but in case of females, they are born with a certain number of oocytes, so there is no de novo synthesis, and the marks are acquired, and they keep adding on. So I think the chances of mitigation in this patrilineal transmission are much higher compared to the matrilineal transmission. About the milk part, whether we have a way to alter the composition of the milk, this is also something which we are studying right now in our mouse study. I don't have, let's say, any conclusive evidence at this point to show that we can alter the microRNA composition of the milk. But what we do know is that by altering the microbiome in the milk, we are able to change their circulating transcriptome. So I think perhaps that kind of intervention, microbiome intervention, could be useful here. But yeah, again, very important questions. Yeah.
Thank you, Dr. Rajab. First of all, this is a cross-pollination of ideas coming from Aga Khan University to King Edward and from Poland to Pakistan. Really grateful. It has taken my mind back to my early days when I was studying Quran. And I just want, there is a comment in the question.
Sure.
The comment is that the sanctity of human breast milk is basically such that when we were forbidden to marry our mothers, our sisters, our father's sisters, and mother's sisters, and nani, dadi. In the same ayah, it was written, وَأُمَّهَاتُكُمُ اللَّاتِي أَرْضَعْنَكُمْ وَأَخَوَاتُكُمْ مِنَ الرِّضَاعِ. And those who became your mothers by having the breast milk, or your sisters who had shared the breast milk. So the question is, in the last part of the last century, it was an era, an emerging evidence of mitochondrial inheritance. Is there any evidence of actual genetic inheritance in the human breast milk?
Yeah, so the evidence is there for this epigenetic transmission, not like the genetic inheritance itself, but if you think about and this reference to the Quran, I think to me all these things we are learning that the predictions from the Quran, they had very strong biological embedding and basis. And if you think about it, this part, just how a mother transfers her genome and her epigenome, to the next generation. Similarly, the lactating mother, the Rozai mother, right? She is also doing the same. Now it is going through epigenetics, but if it is contributing to changes in the genomic regulation of the offspring, to changes in their behavior, then it makes perfect sense that this is something which is in parallel, and this is something which Quran could predict and even guide us years before. So the evidence is there for this epigenetic transmission. Now, in terms of the mitochondrial transmission, I think that has more relevance to this germline-based transmission, because in germline, the mitochondria are contributed by the mother and not from the male germline. So I think that there it's clear that any kind of mitochondrial disease is through the mother. But in the breast milk, it's mainly epigenetic transmission. I think the first question, it's very pertinent, but again, just like how milk, transfers immunoglobulins and all these other factors, I mean, it's not really surprising that the epigenetic factors induced by trauma can also be passed through milk. At this point, and this is where I emphasized, we have very strong correlative evidence, right? But what we don't know is whether these children who have changes in their social and adaptive behaviors, if they will also develop a pathology. If we see clearly by longitudinally following them up that they will develop, autism, ADHD, or any other kind of neuropsychiatric pathology, then I think we can give a recommendation like that. At this point, what I would clearly say is that the breast milk is not the problem, trauma is the problem, right? So what we need to have is that for women who are planning to conceive and they have had significant traumatic experiences, which unfortunately are very common in our society, I think it's important for them to work on their mental health. It's also important for their husbands, the families, and everyone to realize how this could eventually impact even the composition of their breast milk and how that could then impact the behavior of the children. So I think the emphasis should be on removing the lingering effects of trauma and improving mental health rather than asking them not to breastfeed. So this is what I would want to emphasize. If in future we see clear indication that pathologies are developing, then we can go for more strict guidelines. About your question about the SOS Children's Village. So in SOS Children's Village, we are looking at a certain form of parental care, which is their model of foster care, right? Now the thing is that we clearly see that compared to their classmates, which was our control group, they go to the same school, they have similar food, socioeconomic, similar recreational facilities, similar education. We clearly saw that there was a lot more depression and anxiety in the children living in the SOS village. Now these were new children, these were new recruits to the SOS village. They are within 1 year of being separated from their mothers. We don't know whether these things will be still there after many years of living there, but definitely we see that separating from the mother and living with a foster mother, it doesn't it doesn't really substitute for that. Like real maternal care from a mother who is not stressed is the best form of nurturing. Thank you very much.
Rahim, it's been a sheer delight to hear Professor Ali Javed. I think I would just like to give credit to Dr. Athar Saeed. As Abdullah mentioned, basically Universal Healthcare Symposium, which is now in its 8th year, has been his brainchild. I call him a bit of an eccentric because he always thinks outside the box. King Edward alumni events have been happening for many years, predominated by our classmates coming from USA. UK has been a relatively recent kid on the block. And I think rather than just doing a conventional knowledge exchange, he wanted to think of, can we generate an idea which was universal health care, basically free health care for everyone in Pakistan. Now, it's again a bit of a paradox because whilst the government, because of all the financial pressures, is moving towards giving up its sort of primary responsibility and focusing on what they can manage within the resources they have, but I think what we are learning from outside world is government is best placed to provide the free care to all of its population, because that's the cheapest and the best care. So that was the one thought I just wanted to disseminate. The second one was in terms of, again, he has been quite creative in thinking that when we invite individuals for Ommaya lecture, we invite very eminent scholars who are from within us, because we again get a bit of sort of influenced by the way West or the Western world is doing research or cutting edge technology and innovations, but it's just inviting people who have been from our own background, who have gone outside, done an amazing work, and they can share their findings. So I think, it's fascinating trying to understand all the neuropsychiatry, but the point I wanted to highlight, especially for our young generation sitting here, and I know most of you have जो कहते हैं ना लाए आए हैं बट कुछ ऐसे होंगे जो लाए गए and I can imagine from the marshal standing on the both sides is probably focusing not obviously focus on the neuropsychiatry bit, but the key message I wanted to give was as we are producing more and more doctors, the traditional clinical practice routes are saturating and they are saturating very fast. So probably worth thinking who was Ommaya, maybe you can read on YouTube what Ali Javed has presented is where does the future lie for you? Is there a future outside traditional clinical medicine? Is there a future for innovative medicine there? Because the world is wide open there and it's just evolving. Neuropsychiatry is something that we're just trying to grapple with. And he has done an amazing job in terms of making it easy for us. So probably just worth thinking, could one of you be working on those footsteps and maybe progressing science further? So that's where I wanted to conclude. And I would like to massively thank the Vice Chancellor Sahib and all of his colleagues and in terms of providing us the platform and going out of the way and making it a very comfortable trip for us every year. It's always an amazing experience to come see colleagues, meet friends. Thank you.
First of all, let me welcome my dear brother Mujtaba, the senior minister, Government of Punjab. I think he has been minister more than half a dozen times and completed the tenure, so he's the most experienced gentleman in the cabinet. He came to our function at KE at a very, very short notice. Thank you so much, Mujtaba, for being here and supporting King Edward. That reflects his love for King Edward Medical University and for the Vice Chancellor also there. I think Ali's lecture was just music to my ears and was a great talk. I will just, me and my group are already working in Pakistan on these things. One of the major studies that we are undertaking right now is looking at the epigenetic factors in diabetes. And that is when we look at Punjab boasting to be the best performing province of the country, the skilled birth attendance in that study was available only to 28% of the pregnancies in Punjab. And the second antenatal was only available to 19% of the pregnant ladies. So that reflects the poor outcomes for the fetus for many reasons. One of the main reasons we think is because of the lack of antenatal care. The genetics do not come into play that much as epigenetics do. And Pakistan now, shamefully I'll say, nothing to be proud of, is titled as world capital of diabetes, because age matched we are registering the highest number in the world, and year 2050 we will be still number one, unfortunately. And one of the factors is the poor environmental conditions in the fetus, especially during the early part and middle part of the development. And we think that messenger RNA and alterations in the RNA of the play a very key role in causation of that diabetes once the child is born 20 to 40 years later. And also all the non-communicable diseases are much, much common in those rather than ladies who got a very good antenatal care. So I think that it would be great to work with him. We are already working with Poland, but McMaster University in Krakow, we are their collaboration. And so I think about the breast milk, again, we are trying to formulate a study that what happens if you are exclusively breastfed for 24 months, and that is what our Quran says about Musa, that Almighty Allah arranged in such a way that he was again fed by the biological mother. So when you are fed by the biological mother or as versus surrogate mother, they may be very, as Ali just very elegantly described, that messenger RNA abnormalities and more inflammation set in, the cytokine storms and things like that. So I think if you look at outcomes of pure biological mother-fed children, they are supposed to be much healthier at the age of 12 years and later on. I think the reason is that the formula milk that usually is started very early, and Sarah knows it more than I do, that very few pregnancies in Pakistan are now exclusively breastfed, because very early on they are being fed mother to some extent, but also they are joined by the formula milks. And the formula milks are very, very toxic to the RNA. And that is the one reason that we think is, and we are putting up a study together to suggest that. So breast milk is, of course, the best thing as promoted by Allah Almighty Himself. So that is one. So another important thing that we are looking at is microbiome changes in the intestine, because we all know GLP-1 and GLP-2 and GIP is produced by small gut cells. K cells, they produce trizepatide, and GLP-1 is, which is GIP, and GLP-1 is produced by the L cells of the. And if the gut is inflamed, then obviously there is no endogenous production of these incretins. So what happens is, don't worry. So what happens is that If you are taking artificial sweeteners, we think in Pakistan that is a big, big culprit, because that alters your microRNA of the gut, which becomes, that's one reason that we should be promoting. After this, we'll be hopefully served a very hefty lunch by my brother Azaz Mahmood, and there will be Diet Cokes there, and everybody will be looking at Diet Cokes. So we think, as little humble scientists, that it's much better to take a full ordinary Coke. That is also notorious, but if you are given a choice, you should be taking that, because the Diet Cokes, they disrupt your microbiome. Totally, that disrupt your endogenous production of these incretins. So, great lecture. I learned really a lot, and we look forward to collaboration with my group. Some members are sitting here also. So, inshallah, maybe next alumni, Allah bless us all, the health and happiness. We will be hopefully coming up with results of those studies that we are conducting right now. Thank you so much, Mahmood Ajaz, for inviting me, for being here, and it's always a pleasure.
Sir, we are now going to officially close our annual scientific session and start with our closing ceremony. I request the Registrar, King Edward Medical University, and Secretary Organizing Committee, Professor Syed Asghar Naqi, to take his seat at the head table. I request Chairman Organizing Committee, Professor Muhammad Imran, to take his seat at the head table. I request Chairman Audiovisual Committee, Professor Muhammad Haroon Hamid, to take the seat at the head table. I request Chairman Scientific Committee, Professor Ali Madi Hashmi, to take the seat at the head table. I request Chairman Refreshment and Entertainment Committee, Dean Basic Sciences, Professor Dr. Tareem Fatma, to take the seat at the head table. So we are all going to stand for national anthem. I'm going to invite Dr. Asif Khan, President, KEMCA-UK, for his brief closing remarks.
Assalamu alaikum. I think I've already shared some presentation yesterday, and I wanted to first of all thank you, Vice Chancellor, all my respected colleagues, my teachers, my colleagues from KEMCA-UK. Thank you very much, and my dear students. Thank you for joining, the scientific symposium, Universal Healthcare Vision of Dr. Athar Said, who started for the last 8 years. And this is what KEMCA-UK is going to bring, inshallah, every year. A lot of wealth of knowledge to you all. जब हम हाउस जॉब कर रहे थे तो हमारे एक लेजेंडरी केमकोलियन प्रोफेसर मुमताज सन मरहूम, उन्होंने कहा था कि you Chemcolians are born to walk on red carpet. And that's what Chemcolians are. Wherever they are in the world, they are the leaders. And alhamdulillah, we have seen that. जहाँ पर केमकाना ने अपनी 50th anniversary, I congratulate Kemkana for doing that. Mashallah, they've been serving across the world, and especially coming back to alma mater, and they've been doing a lot of infrastructure work. KEMCA-UK has celebrated last year Silver Jubilee, and this is now, KEMCA-UK is more focused to transfer of knowledge, bringing universal healthcare, because we wanted you guys to develop that specific genetic skills, which not only going to help you, going abroad. I think अगर आप उन genetic skills को, इस knowledge को आप adapt करें, तो आप पाकिस्तान में भी रह के बहुत बेहतर फिजिशियन, बल्कि हमसे बेहतर फिजिशियन बन सकते हैं, क्योंकि आपके पास यहाँ पर इतना ज़्यादा पेशेंट वर्कलोड है, इतने ज़्यादा पैथोलॉजीज हैं, एंड यू लर्न अ लॉट. जो चीज़ें मैंने यहाँ पर हाउस जॉब में सीखी थी, अभी तक जाके वो पैथोलॉजीज मैंने जाके यूके में पिछले 20 सालों में नहीं देखी, एंड दैट क्लिनिकल नॉलेज हेल्प सो मच. हमने यहाँ पर पिछले कई सालों में प्राइमरी पीसीआई जो कार्डियोलॉजी इंटरवेंशन है, जो कि अब माशाल्लाह डॉक्टर जावेद अकरम की सरबराही में जब शुरू हुई लाहौर में पिछले 3 सालों में, वी टेक प्राइड एज ए केमका यूके टीम के जो कार्डियोलॉजी डॉक्टर तारिक अजीम ने यहाँ आके लेक्चर्स दिए और शुरू की. और आज सुबह प्राइमरी प्रिवेंशन के लेक्चर में डॉक्टर बिलाल मोहिउद्दीन जो कि पंजाब इंस्टीट्यूट कार्डियोलॉजी के चेयरपर्सन हैं, उन्होंने आके बताया कि पंजाब इस वक्त लाहौर ओनली 6000 प्राइमरी पीसीआई इस वक्त कंडक्ट कर रहा है, विच इज़ अ बिग आई विल से क्रेडिट टू पंजाब गवर्नमेंट एज वेल पंजाब इंस्टीट्यूट कार्डियोलॉजी एंड जावेद अकरम साहब. वरना आप इससे पहले, क्योंकि आपका हार्ट अटैक अगर 2 घंटे के अंदर फिक्स नहीं हो रहा, योर हार्ट मसल विल डाई, एंड इट्स अ ग्रेट एफर्ट बाय द गवर्नमेंट नाउ दैट आप स्पेशली पंजाब में और लाहौर में ये फैसिलिटी अवेलेबल है. केमका यूके, आई थिंक apart from what we are doing already in terms of transferring the knowledge and everything, we will be starting few more projects inshallah. उसमें से need based scholarship program for our graduates, it has already been started now with the class of 1982. So inshallah we will select our students who needs more support financial, and we will inshallah continue to support every year on long term basis inshallah. In terms of the charity work, because हमारा focus केमका यूके का एक charity का chapter है पूरा जो हम इसको करते हैं, और last 2 years में केमका यूके ने more than 2 crore rupees has been raised for Gaza. So peoples in Palestine, हमारे General Secretary Abdullah Gujar has been leading the project, and last 2 years we are only not supporting them where they are based in Gaza, we also have supported them even during Ramadan. So alhamdulillah we will continue to support that. और यही बात है कि ये केमकोलियन चाहे दुनिया में ना सिर्फ एजुकेशन क्लिनिकल एस्पेक्ट पे काम करें, बल्कि ह्यूमैनिटेरियन क्राइसिस जहाँ पर भी आती है, पाकिस्तान में फ्लड्स आते हैं, हमारी टीम यहाँ पर हर जगह, पंजाब, चित्राल, खुजदार में हमने वी हैव कंडक्टेड अ फुल केमका यूके विलेज, जिसमें पूरे लास्ट 2 years से वी बीन सपोर्टिंग देम इवन इन ईद एंड कुर्बानी. सो इंशाल्लाह वी विल कंटिन्यू टू सपोर्ट इंशाल्लाह एंड सॉर्ट ऑफ दीज प्रोजेक्ट्स. लास्ट नॉट द लीस्ट, लेडी एचएसएन हॉस्पिटल जो कि हमारा नेबरिंग हॉस्पिटल है, उसमें 2 ऑपरेशन थिएटर जिनको हम इंशाल्लाह फुली फंक्शनल कर रहे हैं बाय प्रोवाइडिंग फुल एनेस्थीसिया मशीन सपोर्ट, और इंशाल्लाह वी विल बी डूइंग opening ceremony for that today. So our Kemkolians have supported for that. So 50 lakh rupees we have raised, and inshallah 2 machines will be installed, and inshallah we'll be doing operations in inshallah Lady Hutchinson Hospital. I think I will not take more than time, but I will say inshallah in future our team will be coming here, and thank you very much for all welcome support we get. We love to come back every year, and inshallah next year there will be another president from KEMCA UK, but I think I will still stick to KEMCA UK inshallah as well. Thank you.
Assalamu alaikum everyone. So, what an excellent lecture by Professor Ali. Thoroughly impressive. And if you think about it, what makes it as an excellent lecture is the lifetime spent in doing excellent work. ऐसा नहीं है कि आप 4 कहीं से articles उठा लें and you can create an impactful lecture. It has to build a story, because the story was the work that has gone into putting this together. और हम excellence celebrate कर रहे थे डॉक्टर उमैया की. So I read about Ommaya first, actually not him, but about Ommaya Reservoir जब मैं steps की तैयारी कर रहा था 2005-06 में, and it didn't click, but eventually when I learned, so brain cancer में एक ही drug होती है और वो उस ज़माने में और वो रिजर्वायर में डाल के दी जाती थी बिकॉज इट डजंट क्रॉस ब्लड ब्रेन बैरियर. So Dr. Ommaya, being a neurosurgeon doing brain surgeries, he was thinking of creative ways on how to deliver that drug to the brain. In America में हम एक जुमला बड़ा यूज करते हैं, you can just do things. Okay? If you think of something, just go ahead and do it. You don't need permissions, you don't need to ask, can this be done, can this not be done. वो बड़ा मशहूर story है मोजार्ट के बारे में. He started writing the whole orchestra's operas at age 13. Somebody came to him, he was 30-something-year-old, he said, can I write operas? He said, no, you cannot. He said, but you started writing at age 13. He said, yes, but I was not going around asking others if I can write an opera. So if you're going to go ask around, then of course you cannot do it. It's not going to happen. You can just do things. But that also brings me to another quote in US that we often mention, which is that we don't do things because they are hard or easy. We do things because they are right. So if you are looking for excellence, you have to do things because they are the right things to do, not because they're going to give you benefit now. What is your immediate benefit out of it? And that's what KEMCA stands for. KEMCA is about achieving that excellence, trying to get to a state where you are doing the best that you can. बड़ा again another quote अंग्रेजी में बड़ा यूज होता है कि when you are committed to something you have to give it your 100%. You do not do things half-hearted. If you Netflix पे अगर आप लोगों के पास access है, you will watch a documentary called Hero Dreams of Sushi. That's the only Michelin 3-star sushi restaurant in the world. बहुत hard है achieve करना. 3-star का मतलब होता है कि it's worth flying into that country just to eat at that restaurant and come back. That's 3-star in Michelin. और वो बताया कहता है कि once I decided I'm gonna be a sushi chef, I will even dreams of new ways of making sushi. कि आप उठते बैठते जागते सोचते उसी चीज़ के बारे में सोचते हैं, and you're trying to bring that excellence to you with that full commitment, 100% commitment of what you're doing. Coming back to that lecture, lastly, it was so amazing to see the pattern कि जो impairment आ रही है, traumatic stress, mouse हैं, they are risk taking, बच्चे हैं, वो pleasure impairment हो रही है, defiance आ रहा है. And a lot of these things मैं काम करता हूँ न्यूरोडिजेनरेटिव डिसऑर्डर्स में, हम डोपामीन पे काम करते हैं, रिसर्च करते हैं. सो हम पार्किंसंस में देखते हैं कि डोपामीन, जिनको पार्किंसंस होती है, लो डोपामीन स्टेट के पेशेंट्स, दे नेवर टेक रिस्क, दे आर ऑलवेज वेरी लॉ अबाइडिंग सिटीजन्स. तो उसका एक पूरा प्रोफाइल हम काम करते हैं. बट दिस साइकिल यू कैन ब्रेक योर सेल्फ बाई चेंजिंग दैट पैटर्न, चेंजिंग द हैबिट. अगर आप अच्छी बात रिसर्च की होती है कि हम एवरेजेस की बात तो करते हैं, बट इफ यू रिमेंबर दोस चार्ट्स यू वर सीइंग, देयर वाज अ लॉन्ग स्प्रेड. तो ऐसा नहीं है कि हर बंदा जिसका लाइफ टाइम का पेरेंटल का ट्रॉमेटिक एक्सपोजर है कि उसने लाजमी इम्पेयर्ड होना ही है. वी टॉक अबाउट एवरेजेस, ऑन एवरेज दैट ग्रुप हैज मच वर्स परफॉर्मेंस, बट दे आर ऑल्सो क्वाइट लो, मच बेटर पीपल इन दैट कोहॉर्ट. व्हाई कांट वी बी दैट ऑफ दैट, राइट? दैट वाज KEMCA अबाउट. मैं जब स्टूडेंट था यहाँ पे, तो हमारे एक टीचर थे एनाटमी के, वो कहा करते थे कि देयर इज ऑलवेज स्पेस ऑन द टॉप, कि आप जो जितनी भी स्ट्रगल नीचे चल रही हो, there is always space on the top, just climb the hill. और ऊपर बड़ी जगह है, नीचे आपको बड़ा crowded लग रहा है कि बड़ी बहस हो रही है, बड़ा मुकाबला है, कैसे मिलेगा वो मिलेगा, but once you get to that excellence, you will become very few and it's very easy to get, and that's what makes KEMCA and KEMCA great. Thank you for being here.
Bismillahirrahmanirrahim. The very honorable chief guest, Mia Mustafa Shujaa Rahman, who is the provincial minister for the finances of Punjab, the real treasurer of Punjab. पंजाब का खजाना जिसके पास है, आज वो खुद हमारे पास है. तो सर आपके आने का यहाँ पे बहुत शुक्रिया. King Edward Medical University is really truly honored and privileged that you accepted our invitation to be here today. So it's really a great pleasure to have you here today. And it's an equally enlightening moment for me that my very own teacher, the provincial minister for health, लोग उनको कहते हैं कि ये पास्ट मिनिस्टर, ऐसा नहीं है. Still both the ministers, उन्होंने स्टेज पर बैठ के, both the health ministers, उन्होंने कहा कि हम अभी भी हेल्थ के बारे में इन्हीं से रहनुमाई हासिल करते हैं. और इसी स्टेज पे खड़े होकर किंग एडवर्ड के पोडियम पर कहा कि he is our teacher. So sir, you are still the health minister, and thank you very much for being here today with us. आपकी बहुत मेहरबानी. You are a famous teacher, a great Capricornian. You have been, a vice chancellor of so many universities, जिसको मैं count भी नहीं कर सकता, but still, King Edward Medical University is his first class. So thank you very much. And it's equally pleasurable for me to actually welcome all these esteemed faculty members, the distinguished guests, the dear Campbellians. लेकिन ये उन सब मिला के ये सारी जो महफिल कल और आज की हमने सजाई है हमारी इतनी इल्यूमिनाई के लिए है because they take out their time to come back to Pakistan. So I am very pleased to welcome representatives from APNA, representatives from KEMCA, representatives from KEMCA-UK, and representatives from the KEMCA-UK. And all their presence are here, and I'm so glad you listened to them, and I'm so glad that they are here today. So, sir, thank you very much for being here today with us. और इसके साथ साथ मेरी बड़ी ख्वाहिश थी कि क्लोजिंग सेरेमनी इसमें प्रोफेसर जो है ना हमारे साथ असद हसन साहब ने तशरीफ फरमाओ बिकॉज ही इज द वाइस चांसलर ऑफ दिस यूनिवर्सिटी एज वेल किंग एडवर्ड के जितने भी पुराने वाइस चांसलर्स हैं वो हमेशा वाइस चांसलर्स ही होते हैं वो कभी रिटायर नहीं होते सो दे आर पार्ट एंड पार्सल ऑफ किंग एडवर्ड सो सर थैंक यू वेरी मच फॉर बीइंग हियर और इस सारे टेल्योर में जो कल 2 दिन हमारे गुजरे उससे पहले 27 प्री कॉन्फ्रेंस वर्कशॉप heavily attended, heavily actually designed to benefit the students and the faculty. और उसके बाद कल से लेके आज तक 2 दिन लगातार yesterday 7 parallel sessions, today 6 parallel sessions, numerous lectures, numerous academic sessions which we had to actually cap all of them. Today's lecture by our dear, you have just listened to him, by Dr. Ali Javed. Thank you very much for being here, enlightening us. It was a befitting lecture, so we are very thankful. और इस सारी बातों के बाद, आपकी alertness, आपकी तवज्जो हासिल करने के बाद, मैं अब आपको सलाम करूँगा. Assalamu alaikum. So thank you very much. And I hope that you have all enjoyed this program which we have put together as much as we enjoyed while we were putting it together, जब हम हमारे रजिस्ट्रार डॉक्टर नकी और डॉक्टर मोइन जो हमारे प्रो वाइस चांसलर हैं, both the Professor Imrans and the whole faculty of the King Edward Medical University, we had multiple joint sessions together. हमने बैठे उसमें हमने बात की, हमने एक दूसरे से मशवरा किया, and we thought that it is going to be really a very pleasurable occasion to have the people from all over the world come down to us at King Edward Medical University and enlighten us. So we are very thankful to the alumni because I call them as the greatest treasure of King Edward Medical University. मैं इसलिए उनको ऐसा कहता हूँ because they are the ones who develop, who basically enhance, and who actually glorify King Edward Medical University, and they've been doing so for the last 165 years. But every December, just like migratory birds, they will just come back to King Edward Medical University. But वो अकेले नहीं आते. They bring with them the wealth and wisdom of their knowledge and experience which they have gained over the last 1 year from the best training centers and teaching centers all over the world. और यहाँ आके हमें सिखाते हैं, हमें पढ़ाते हैं, हमें बताते हैं कि जो है ना नया नॉलेज क्या है और हमें आगे कैसे बढ़ना है. So I am so grateful for the alumni to basically come over here in Pakistan and actually help us out in our own endeavor towards जिसके बारे में दानिश बात कर रहे थे, excellence के बारे में बात कर रहे थे. और being a vice chancellor, मेरा सिर्फ एक ही काम है इस यूनिवर्सिटी के अंदर. मेरा काम है to just make sure that this university is actually rebuilt, restored and revitalized and repositioned as the #1 university of Pakistan, and then gradually going ahead and ahead. और इसके लिए विजन मेरा बड़ा ही सिंपल है कि एक तो है कि ये लर्नर्ड सेंटर होना चाहिए कि जो आप लोग हैं इनके ऊपर हमारा सारा फोकस होना चाहिए, एंड दे शुड बी डिस्कवरी ड्रिवन. हमें नया नॉलेज क्रिएट करने की ज़रूरत है, एंड वी शुड बी ग्लोबली इंगेज. जो जिस हम ये इंगेजमेंट्स करते हैं अपनी ऑल ओवर द ग्लोब, ये हमारा काम है इसको करना. एंड आई एम सो प्राउड that the brilliant students of King Edward Medical University, their scholarly faculty, and it's very able administration, they have collectively put this vision together, and now we are on the verge of breaking our steps into the future. Rather, we are there. और मैं आपको 5-6 छोटी-छोटी चीज़ें गिनाऊंगा जो हमने भी recently achieve की with the help of everybody जिसका मैंने mention किया है. First of all, the development of the School of Artificial Intelligence. It is there, it is robust, it is working, and it is in full speed. बहुत अच्छा काम कर रहे हैं और हमारे students ने, sir, they have created 9 apps और जो हमने उसको मार्केट कर रहे हैं, अब प्रोफेसर सायरा अफजल उसके रुए रवान थी, उन्होंने इसको किया, अब इसको डॉक्टर जो है ना उज़ैर उसको लुक आफ्टर करते हैं, बट दे आर गोइंग एट ग्रेट स्पीड और बहुत ज़बरदस्त काम कर रहे हैं। उसके साथ साथ हमारे डिपार्टमेंट ऑफ पैथोलॉजी ने प्रोफेसर फायजा और प्रोफेसर सजाद इज सिटिंग हियर, they were very instrumental in helping me out with the collaboration of the University of Columbia to develop our own biobank. जिस यूनिवर्सिटी का अपना बायोबैंक नहीं, वो कैसे रिसर्च का दावा कर सकती है, वो कैसे कह सकती है कि हम कर सकते हैं। और बायोबैंक का होना तो सिर्फ सफिशिएंट नहीं है, वी नीडेड टू हैव अ जीनोम सीक्वेंसिंग सेंटर, सो वी डिड डेवलप अ जीनोम सीक्वेंसिंग सेंटर, वी हैव अ जीनोम सीक्वेंसर्स वेयर वी एक्चुअली ट्राई टू ब्रेक द कोड ऑफ द जेनेटिक डिजीजेस, द हेरिडिटरी डिसऑर्डर्स, द कैंसर्स विच वी कम अक्रॉस, और उन कैंसर्स की हम जेनेटिक कोड्स को ब्रेक करके वी फाइंड आउट द डिफेक्टिव जीन एंड वी ट्राई टू प्रोवाइड द बेस्ट पॉसिबल एडवाइस and the treatment for these patients. So I'm so thankful for the oncology department, the pathology department. They've been very instrumental in actually putting this thing together. और उसके बाद जैसे जैसे हम ये करते गए, we got our journal as an impact factor ranked journal, the first journal in the public medical universities in Pakistan, apart from UHS, जो कि प्रोफेसर जावेद अकरम साहब ने उसको किया। और उसके बाद शायद अब हम उनसे आगे निकलते चले जा रहे हैं, क्योंकि इस तरह का विजनरी शायद ही किसी और यूनिवर्सिटी को नसीब हो, लेकिन हमें हमारे पास है, क्योंकि इनका एडवाइस हमारे साथ हर वक्त शामिल होता है। सो सर, आई एम सो थैंकफुल टू यू, आपने हमें सब चीज़ों में मदद की। और उसके बाद फिर हमने अपनी QS रैंकिंग को बड़ा किया, उसमें प्रोफेसर सायरा अफजल का और हमारे QC डिपार्टमेंट का बहुत ज़बरदस्त हाथ है। और उसके बाद नाउ वी आर स्टेपिंग इनटू इनोवेशंस। हमारी फैकल्टी ने, हमारी इल्यूमिनाई ने अपने 17 पेटेंट्स डेवलप किए। द 18th विच वाज द लेटेस्ट वाज डेवलप्ड बाय प्रोफेसर इरफान मुनीर, हु इज अ केमकोलियन। सारी रिसर्च उसकी यहाँ पे कंडक्ट की गई एंड वी हैव डेवलप्ड आवर ओन ऑटोमेटेड पेरिटोनियल डायलिसिस मशीन, जिसको FDA ने अप्रूव किया, उसको पेटेंट किया, एंड नाउ इसकी सेकंड जनरेशन जो है ना मार्केट हो रही है। दिस हैपेंड ड्यूरिंग दिस ईयर एंड लास्ट ईयर। सो आई एम वेरी ग्रेटफुल फॉर द एल्यूमिनाई टू हेल्पिंग अस अ लेंडिंग हैंड। दैट इज़ हाउ द यूनिवर्सिटीज़ एंड द एल्यूमिनाई दे वर्क। अब ये बहुत ज़्यादा डोनेट करते हैं, ये इतना ज़्यादा पैसा हमें देते हैं कि हमें किसी आगे के आगे हाथ फैलाने की ज़रूरत ही नहीं रहती। मतलब दी KEMCANA and the KEMCA and the KEMCA Pakistan, they donate so much कि हमें ज़रूरत नहीं है कि हम किसी के आगे से कहें कि हमें कोई चीज़ लेकर दे। बट उसके अलावा जो ये हमारी मदद करते हैं, that is the academics which they build, that is the knowledge which they impart, that is the research endeavors which they actually sponsor. So once again, मैं इनका शुक्र अदा कर करके थक जाऊँगा, लेकिन शुक्रिया पूरा अदा नहीं हो सकता। लेकिन मैं इनकी तरफ से अपने स्टूडेंट्स को वही पैगाम देना चाहता हूं जो मैं हमेशा से देता आया हूं, कि देखें बात यह है, इट्स टाइम टू एक्चुअली मेक श्योर दैट ये आप मेडिकल कॉलेज में पहुंच कर डॉक्टर्स बन रहे हैं, ये यहाँ पे बात ख़त्म नहीं हो जाएगी। ऑलवेज सीक न्यू नॉलेज, बिकॉज दिस न्यू नॉलेज इज गोइंग टू ब्रिंग यू एवरीथिंग। Enter into genomics, enter into gene sequencing, enter into robotics, enter into artificial intelligence, and this is going to bring you everything, whatever you want in your life. Tamam izzat, shohrat, नेक नामी, पैसा कह ले, everything will be gotten by you. लेकिन जो दानिश की बात मैं यही रिपीट करूंगा कि seeking new knowledge is not enough. You have to seek excellence. You have to seek excellence, because excellence is also going to make sure, और जिस हमने कहा कि it is a habit. ये ऐसा नहीं है, it is not going to come from one day. It is a habit.
Lifelong learning and ethical medical practice. Your contribution will shape the future of healthcare in Pakistan. I also acknowledge the invaluable role of faculty members and mentors who continue to guide and nurture the next generation of healthcare leaders. Before I conclude, let me reaffirm that the Government of Punjab stands firmly with the medical community. We recognize and respect your sacrifices and are determined to create an environment where doctors can serve with dignity, security, and professional satisfaction. I congratulate King Edward Medical University on the successful organization of this landmark international conference and extend my best wishes for the scientific session, workshops, and deliberations. I sincerely hope that the outcome of this conference will translate into improved patient care and a strong public health system. Thank you.
Guest. For this, I'm going to invite Vice Chancellor, King Edward Medical University, Professor Dr. Mahmood Ayaz, to please come for presentation of shields. I request Chief Guest, Mr. Mushtaba Shajaur Rahman, to please come for presentation of shields. Award. To present this award, I'm going to request President KEMCA, Dr. Danish Bhatti, to present citation of Dr. Muhammad Fateh Shahzad, recipient of Lifetime Achievement.
Assalamu alaikum everyone. It's my pleasure to invite Dr. Fateh Shahzad for this Lifetime Achievement for KEMCA. As you know, it takes a lifetime to really build an excellence, and among many people who have done so much work for KEMCA, it is my pleasure to invite Fateh Shahzad. He was born in Bhavalnagar, graduated KE in '88, and has done residency in internal medicine in Columbia University, and now practices in Danville, Kentucky. And he has served KEMCA as treasurer, as secretary, as president-elect, and president of Sun Foundation. Fateh bhai ko aksar log jante honge because he is the power behind bringing Chemcolians to US, growing the KEMCA presence and influence in North America, and really making our family more robust. He has also given a lot of time back to infrastructure projects in Pakistan, developing many initiatives for Mayo Hospital, including pediatric burn unit, ventilators and other medical equipment, and building of KEMCA village for victims flood in rural Sindh. And as you know, he has served also in many committees in Apna, but in KEMCA his achievements are second to none. So it's my great pleasure to award him the Lifetime Achievement for KEMCA. Thank you so much.
ये जो केमकोलियंस बैठे हुए इनका लगता है अमेरिका जाने का कोई इरादा नहीं है। जो तालियां जो मुझे बता रही हैं कि वो इतने मतलब कि फतेह के लिए आप तालियां बजाएंगे तो ये ले जाएंगे आपको।
Thank you. Our next award is KEMCA Dr. Masood Akbar Research Award for KEMU undergraduate and postgraduate students. In the first category, we have postgraduate recipient. The first prize for this is for Dr. Waleed Tariq, resident thoracic surgery in the Department of Thoracic Surgery, King Edward Medical University, for rupees 50,000. Dr. Waleed Tariq. Second prize of 25,000 rupees for Mariam Ayub, Fellow Child and Adolescent Psychiatry, King Edward Medical University. Third prize, rupees 25,000 rupees for Dr. Sania Mumtaz. Dr. Sania Mumtaz, Fellow Child and Adolescent Psychiatry, third prize. For our undergraduate recipient, we have Muhammad Nabil Siddiq, fourth year MBBS. Dr. Nazish is going to receive the award for Sania Mumtaz. Undergraduate recipient Muhammad Nabil Siddiq, first prize rupees 50,000 rupees, fourth year MBBS student. Second prize Shamika Chima, second year MBBS, 25,000 rupees. Third prize Fatma Shahid, fourth year MBBS, 25,000 rupees. Fatma है? Fatma. Fatma Shahid, 3rd prize, 4th year MBBS. Now we have KEMCA-UK need-based scholarship for 10 undergraduate students. This Dr. Asif Khan, President, KEMCA-UK, will present this to our Vice Chancellor, Professor Dr. Mahmood Ayaz. Okay. So I will request Vice Chancellor Saab to present shield to our faculty members who have actively participated in this conference. Professor Dr. Mohammad Moin, Pro Vice Chancellor and Convener, 45th Annual Conference, International Conference. Professor Dr. Muhammad Imran, Dean, Medicine and Allied Specialities, Chairman, Organizing Committee. Professor Dr. Syed Asghar Naqi, Registrar, King Edward Medical University and Secretary, International Annual Conference. Professor Dr. Ali Madi Hashmi, Chairman, Department of Psychiatry and Chairman, Scientific Committee. Professor Dr. Ibrar Ashraf, Chairperson, Department of Surgery and Chairman, Exhibition Committee. Professor
Professor Dr. Muhammad Harun Hamid, Chairman Department of Pediatrics and Chairman Audiovisual Committee for 45th KEMCON. Professor Dr. Tahreem Fatma, Chairperson Department of Anatomy and Chairperson Refreshment and Entertainment Committee for 45th KEMCON.
Professor Dr. Nazish Imran, Chairperson, Department of Child and Family Psychiatry, Chairperson, Publication Committee. Professor Dr. Uzair Ahmed, Head of West Surgical Ward, Chairman, Hall Management Committee. Professor Dr. Mustahsan Bashir, Chairman, Department of Plastic Surgery. Professor Dr. Faiza Bashir, Chairperson, Department of Pathology, Chairperson, Reception Committee. Professor Dr. Nadia Khurshid, Head of Ops and Gynae, Co-Chair, Reception Committee. Professor Dr. Imran Hassan, Professor of Medicine, Chairman, Refreshment and Entertainment Committee. Professor Dr. Munib Ashraf, Chairman, Department of Pharmacology, Co-Chair, Refreshment and Entertainment Committee. State Secretary, closing ceremony. Dr. Muhammad Umar Baraj, Assistant Professor of Surgery, Chairman, Transport and Accommodation Committee. Mohammad Rashid Javed, Assistant Registrar and Organizer, International Organizing Committee. Now I will request our chief guest to present a shield to our Professor Dr. Mahmood Ayaz, Patron-in-Chief and Vice-Chancellor, King Edward Medical University. Now I will request Vice-Chancellor, King Edward Medical University to present a memento to our chief guest, Mr. Mushtaba Shujaur Rahman. Now our Vice-Chancellor Saab will present shields to our honorable speakers and guests. Shield for Professor Ali Javed. Dr. Asif Khan. Dr. Asif Khan, Dr. Atar Saeed, Dr. Tabinda Dogal, Dr. Ambreen Rauf, Dr. Dr. Abdullah Gujar. Dr. Tariq Azim. Dr. Adeem Murtaza Raja. Dr. Fatima Asif. Dr. Sajjad. Professor Dr. Asad Aslam. Dr. Javed Akram. I will request Dr. Sajjad, Professor, Pathology Department, to please come. We have a shield for you. In the end, we have two very special shields for Ahmad Rashid, Student League, 45th KEMU Annual International Scientific Conference. Radia Wahid, Student League, 45th KEMU Annual International Scientific Conference. Thank you so much, sir. Thank you, sir. But for the vote of thanks, I will invite Dr. Moin.
I'd like to thank everybody for attending this momentous occasion for Alumni KEMU-Con 2025, and I'd like to thank the Minister for Finance for joining us, Professor Javed Akram, Professor Asad Aslam Khan for joining the occasion, and obviously the team from KEMCANA, Dr. Danish Bhatti, Dr. Asif for joining the sessions over here. I think the sessions were very well attended, and the working for the meeting did not end over here. It was something which went on. There were unsung heroes of the event, which I think Mohsin is one of the persons. He works in KEMCANA. He deserves a big, he was sort of working late night till 3 p.m. Rashid is another person who worked very hard. The IT team over here, the projectionist over here, and the photography team. But not the least of them all is the faculty who organized the organizing committee for this KEMCANA event. I think they worked very hard. The psychiatry department, the chairman scientific committee, Professor Ali Madi Hashmi was there with us every day. His team, all the residents were here and working with us, developing the scientific program and sort of formulating the program. And it was a sort of a very combined effort from all of us, the chairman, organizing committee, Professor Muhammad Imran, and the secretary organizing committee, the registrar. And I think the best credit goes to our vice chancellor, Professor Mahmood Ayaz. I think his thirst and his for knowledge and for excellence never finishes. That's why all the time he comes in and he says that you have to strive that, because this is something which we want to inculcate in all the Chemcolians who are here. And we look forward to seeing you next year, inshallah, at KEMU-Con 2026. That will be going to be held in March, I think, next year. Thank you very much, everyone, for joining us.